Welcome to the Veeda Lifesciences podcast series, where scientists, business leaders, and clinical researchers discuss the latest and most critical topics in drug development. I am Bhavika Maheshwari, and my esteemed guest for today is Dr. Pritesh Contractor. He's the Deputy General Manager from our Bioanalytical department. He serves as a scientific and technical lead for biopharmaceutical companies. Today, Dr. Pritesh is bringing over 19 years of experience in regulated bioanalysis, specializing in developing, validating, and applying bioanalytical methods to support clinical studies for both generic and new chemical entities. He also has extensive experience in conducting elemental analysis, where he's focusing on various iron formulations such as iron sucrose, iron dextrin, and ferric carboxymaltose, as well as other compounds like potassium chloride and zinc sulfate. These analyses are particularly when the element is derived from pharmaceutical products. In this episode, we would be diving into an exciting discussion about ferric carboxymaltose or FCM, a molecule that has been transforming how we are managing iron deficiency in anemia. Welcome, Dr. Pritesh.
Thank you so much, Bhavika, for this nice introductions. And I'm really excited to talk about this very important topic on this FCM and our experience on this development and molecules validation.
Let's start with the basics. What makes FCM such a breakthrough in managing iron deficiency in anemia?
Yes, Bhavika, it's a great start to discuss this FCM journey. At its core, FCM is a remarkable because of its unique chemistry. It's a ferric iron bound with the carboxy maltose. FCM is an intravenous iron preparation used to treat a iron deficiency in the anemia. It's often prescribed when the oral iron supplements are not effectively working or not tolerated to the patients, or when a rapid increase in the iron is necessary. So that's why this useful for the patients.
That sounds fascinating. Who are the patients that are primarily getting the benefit from this therapy?
Okay, for FCM, as I mentioned, the patients who has the deficiency of anemia, specifically with the patients with the chronic kidney disease on hemodialysis, or a pregnant woman struggling with a severe iron deficiency anemia, and any of the one needing the rapid, reliable iron replenishments when the oral solutions are not working. When administered intravenously, the ferric carboxymaltose bypass the GI tract. So it is useful for the patients who cannot absorb the iron effectively from the oral dose. Maybe it's due to the chronic disease or due to some malabsorptions of the patient's medical history. So for those patients, these IV formulations are very much important. Also, the carboxymaltose complex is very stable in the bloodstream, and it's allow for a slow and a controlled release iron of the ferric iron.
It's impressive how it bypasses the hurdles of oral iron, going straight into the bloodstream. But Dr. Pritesh, how does Vida Life Sciences add value to this therapy?
Each aspect of our analytical techniques aims to set a new standards for the industry. For instance, we were the first in India. Yeah, we are in the first in India to develop a bioanalytical method of this FCM. This is the ferric carboxymaltose-associated iron in the serum by using a unique technique of HEC ICP-MS technique.
Speaking of the regulatory guidelines, the FDA has some rigorous requirements for measuring FCM. How does Vida Life Sciences tackle those?
Recently, in the November 2024, FDA revised its guidance for this FCM. And as per this revised guidance, they suggested two options. Option A is the measurement of ferric carboxymaltose-associated iron in serum, and the bioequivalence is based on the 90% CI of this FAI. And they also suggest the option 2, it's option B, which is the measurements of two different component. One is the total iron in serum, and second one is the transferrin bound iron in serum. And the bioequivalence is based on the 90% CI, where the maximum value of the difference in the concentrations between the total iron and TBI is measured over the all time points. And secondly, the difference in the AUC, area under the curve, is measured for TOI and TBI. So that's option 2.
I would like to start off with option A, evaluating ferric carboxymaltose associated iron in serum. Could you share how Veeda Life Sciences is tackling this approach?
Yes, of course, Bhavika. Let me walk you through our process step by step. To dive into this, we use a combination of size exclusion chromatography, which is SEC, and it's coupled with the inductively coupled plasma mass spectrometry. It's ICP-MS. Now let me explain why we choose these tools, because in SEC, as a filter that separates a serum protein and the iron complexes based on their molecular size. It's perfect for isolating the FCM bound iron from the rest of the serum components. Once we have separated everything on then it's ICP-MS in steps in. It's incredibly sensitive and precise, ideal for the detecting and quantifying the even the tiniest trace elements of this iron. So with these combinations, we are doing this analysis of FAI. Using this method, we could clearly differentiating between the iron that's are bounded with the formulation, or the we can say it is a formulation bound iron, or we can say it's a FCM, and the iron naturally present in the serum. So the iron which is naturally present in the serum, which is known as the transferrin bound iron, it's a TBI. So that too we have to separate by using this SEC chromatography. And these outcomes tells us that the FCM released iron into the circulation is a controlled, steady way, and which is exactly what you want to be safety and efficacy. So this approach allows us to map out iron distributions in a detail and give us insights into how the FCM behaves in the body. Beyond that, it's a great tool for the monitoring iron therapies, especially for the patients with the iron deficiency. Now, when we validate the method, we set the linearity range for the ferric carboxymaltose in the range of 10 microgram to 600 microgram based on the anticipated Cmax values for this molecule, which is in between to the 300 to about 350 or 400 somewhere. Now, these developed method we have also implemented for one of our exploratory study, where we measure the FAI, and in the same samples we also measure the total iron and transferrin bound iron. So the results are compared in between all these three. The results were very close, and when we substitute this TOI minus TBI value, and what we observed that it is perfectly comparable with the FA analysis. And the differences between these two experimental and the theoretical values is less than 20%.
That's fascinating, Dr. Pritesh. The level of precision you described is impressive. I am curious to understand, how did Vida Life Sciences handle option B, where both total iron TOI and transferrin bound iron TBI were evaluated? What challenges did you encounter and how did you overcome it?
Okay, as mentioned in the option B in the USFDA guidance also. So in option B, they suggested to measure the two separate component. One is the total iron in serum, and the second one is the transferrin bound iron in the serum. And these both required a separate method development and the validations. For total iron, there will be no any exhaustive trials are required. But yes, for transferrin bound iron, we have to do some trials to isolate the transferrin bound iron from the drug bound iron as well as the any possible free content of the iron in the serum samples. So we tested a various solid phase extractions columns to optimize this process. And after extensive evaluations, we finalized one of the specific extraction cartridges containing the 1 gram of the pre-filled content with the 6 ml capacity. And this setup allows us to reliably isolate the TBI. The linearity range is also one of the important considering the Cmax. So for total iron, as the Cmax is very higher, so the linearity range is 0.4 microgram to 800 microgram. And for TBI, the concentration is very limited and in the very small or narrow range, so which is giving about 7 to 8 microgram of Cmax. So therefore, the linearity range for TBI is about 0.4 microgram to 16 microgram only. Now, to validate this method, we prepared a formulation spiked quality control samples at both higher and a lower concentration level, specifically for the TBA. So, this confirms us the reliable measurements of the TBI. One of the experiment was proven during the validations called a extraction cartridges efficiency experiment. With this experiments, we are saying that there will be no any unwanted iron measures for the accurate measurements of the TBI. So that part also we are carefully considered during the method development and validation and implemented during our study sample analysis also. One of the most innovative aspect was optimizing the AUC, the calculations of the TOI and TBI. Since the FDA guidelines emphasize using all available data points, we ensured no baseline corrections were applied. This approach not only met regulatory requirement but also improved the robustness of our analysis. While the optimizing process was complex, the results were worth it. We achieved a high degree of accurate repeatability and ensuring our bioequivalence assessment under option B stood up the FDA's rigorous standards. So with this, we developed the method, validate the method, and the same validated method implemented for the study sample analysis also.
It's fascinating, Dr. Pritesh. The level of precision you described is impressive. There's no doubt that a lot of effort indeed goes into ensuring precision. What challenges have we faced in developing these methods? Can you please elaborate on that?
Yes, yes, Bhavika. Of course, when we talks about the challenges are part of the journey, and I firmly believe that in drug development, challenges are not just roadblocks; they are opportunities to innovate. At Veeda Life Science, we don't shy away from the complex; we thrives on it. So let me break it down for you and what challenges we are observed during the method development validation. First of all is separation of the various iron species, including the drug-bound iron, free radical irons, and the transferrin-bound iron in serum or plasma samples when using the size exclusion chromatography coupled with the LC ICP-MS, because it is one of the core requirement. If the separation is not achieved, then our quantitations and measurement is not perfect. So we definitely confirm and provide the data for the separations of the each and every iron component present into the serum or a plasma, so that we take care during our method development phase for this size exclusion chromatography. Second one is the careful optimization of the mobile phase, size exclusion, analytical column, and a reconstitution solutions compositions is essential for the developing a robust and a reproducible method, because these are the main or key parameters which are directly affecting on your separations, resolutions of the chromatography, and the data's response and the results at the LLOQ level. So we have seen some challenges during the development, and after solving one by one all these challenges, we fix our mobile phase column, its length, then the composition of the reconstitution solutions, and finally it is met with all acceptance criteria. Now the third one is the precise selections of the autosample rinsing solutions or a reagent, because as we know, the concentrations of total iron is very high. It's about 300 500 microgram. So when we are talking about the size exclusion chromatography, we need to remove this carryover generated from the equipment. So the selections of these autosample rinsing solutions are very very important to remove the entire flow lines, to avoid any addition from the preceding sample. So that selection is also very very important, that we observed during the method development, and carefully considerations of this rinsing solutions and reagent, we fix these all parameters. And the last one is one of the important part for considering the nature of the samples. It's a hemolysis nature of the samples. So as we know, the hemolysis nature of the sample serum or plasma is affecting the quantitation of the total iron and transferrin-bound iron. So this needs to be identified properly and recorded appropriately, because the hemolysis samples will affect the analysis of total iron or transferrin-bound iron. It's equally affecting the analysis of FAI also. So these samples are not analyzed during the study sample analysis, but the necessary tastings we did during our development and validation section.
Clear that Vida Life Sciences' dedication to innovation is making significant strides. Before we wrap up, what's your message to our sponsors considering FCM studies?
Yes, Bhavika. Veeda Life Science is your ideal partner for FCM studies because of our pioneer methods, advanced instrumentations, and a proven expertise. We deliver data that sponsor can trust, data that supports successful submissions, and a better patient's outcome. With Vida Life Science, you are not just getting a service, you are gaining a partner who committed to achieving excellence in everything that we do. So that's message from me to our audience.
Thank you, Dr. Pritesh, for sharing such valuable insights, and thank you to our listeners for joining us today. Stay tuned for more episodes of Vida Life Sciences podcast series, where we are going to explore cutting edge innovations in drug development. Until next time, take care. Thank you, everyone.
Thank you. Thank you so much.